In progress.
You switch off your cell phones, please. I'm going to wait to see action. We have a very high-tech center here, which has special devices that go up the moment you don't switch it on. So be warned. Welcome to the first public lecture at Chinta. Chinta, as the word suggests in Bengali, means the thought, an idea, and the idea in this case is perhaps a little out of the box, a little outrageous, a little daring. That is of creating a cutting-edge center for research in translation of neuroscience. And so it's enabled by the vision and dreams of Dr. the Chatterjee and enabled by this fantastic team. And we just got started a couple of months ago, and we're part of a larger organization called TCG CREST, the Center for Research in Technology, other centers under this umbrella. And today I'm delighted to have you all here on this public lecture. To be given by somebody who's truly an iconic and transformational figure in neuroscience. Richard Morris is a professor of neuroscience in Edinburgh and was until recently director of the Center for Cognitive Behavioral Systems. He read natural sciences at Cambridge and did his DPhil at the University of Sussex. His early career included a period helping to build an exhibition at the Natural History Museum and working for BBC Television before taking up a lectureship in Scotland. He remains active in university teaching, with his long-standing research interest being the neurobiology of cognition, particularly the role of synaptic plasticity in memory formation. His pioneering research has elucidated many aspects of memory formation and mediating this, the neural mechanisms of memory encoding and memory retention. Beyond the lab, he set up Edinburgh Neuroscience in 2005, jointly with clinical neurologist Charles Warlow, which they directed together for the first 5 years. Outside Edinburgh, he served as head of neuroscience and mental health at Walkinshaw in 2007 to 2010, where he helped set up the new Sainsbury Walkinshaw for neurosurface and behavior at the University College London. He was a co-recipient with prominent physiologists Tim Bliss and Graham Collinbridge of the Brain Prize awarded by the Newbeck Foundation, which are the most prestigious international recognition in neuroscience. He was also elected a Fellow of the Royal Society in 1997, the Academy of Medical Sciences in 1999, the Norwegian Academy of Sciences and Letters in 2007, and more recently of the National Academy of Sciences USA. He was also appointed commander of the British Empire in 2007. Now, there are many famous people. Some of them are famous for being famous. Some have big citations, big papers, big egos, and neuroscience have plenty of all of them. But every now and then, there's a figure who's truly transformational, not because of citations and impact factors, but because of their scholarly and detailed science into results that come out in a rapidly growing field. So as we get taken over and taken doomed by technology and the flashy findings, there are a few people who need to take a step back and think of it differently, and provide insights that are otherwise missing. And it's in that sense that Pokemon is truly exceptional. But I've had the fortune of interacting with them for a couple of decades. Every conversation has come away completely blown away by how we can look at something completely different and provide new insight. And so in that sense, for a very, very young institute like ours, I think it's extremely fortunate that he's chosen to come here and beginning in India. He's part of a wild society and we really have the choice of doing brand that else these years. So I'm deeply grateful for that, and we've joined in with the region.
Well, Shona, thank you very much, and thank you to all of you for coming on a Good Friday. Perhaps it's a holiday here, I don't know, but I greatly appreciate it. I know that one or two of you have come some distance. This lecture is a sort of your guinea pigs for a lecture I have to give in the Royal Society in about 2 weeks' time called the Ferrier Lecture. It's one of the annual lectures. So any comments or criticisms will be gratefully received afterwards. And I can then try to fine tune it. And I want to say it's a great honor to be giving it here in Calcutta, which I understand was the capital of the British Raj until I think 1911 or so. I know I look young, but I was actually born in June 1948. So I'm not quite sure what my parents were doing after August 1947, but you can think of me as the product of their celebration of Indian independence. So the theme of my talk, as the title implies, has to do with the making, keeping, and losing of memory. And in doing that, I want to talk about the various brain regions and activity patterns that mediate the encoding of information into memory. Then some of the selection mechanisms that determine what we keep and what we lose. And then finally, under losing, forgetting, which can be both benign and more serious. And the latter, of course, is portent perhaps of things that may be to come. Now, I'm very grateful to lots of people who I've had the privilege to work with over the years. Got various sorts of lab photos I could show you. I'll just share one only. They've been a great crowd, and I'm just the sort of spokesperson for them. So please understand that if I fall into saying I observed something, what I actually mean is we observed something. And if I rise to saying we observed, I mean that they did it. So who was David Ferrier? David Ferrier was a renowned 19th century neurologist and experimentalist. He worked as a physician, but also in the laboratory, and he was best known for introducing and refining the technique of electrically stimulating the brain and seeing the consequences of doing that. And that led to him supporting the idea of the notion of localization of brain function. He was elected to fellowship of the Royal Society at a very early age, and one of a group of people who set up now acclaimed journal of brain. Now, others have used the same technique. Wilder Penfield in Canada, the celebrated 20th century neurosurgeon, who also used the technique of electrical stimulation. And this helped to refine the idea of a homunculus across the surface of the motor and somatosensory cortex, in which the amount of area devoted to a particular body part was in proportion to its importance. So the hand area is very much larger than the back area, that kind of thing. And you get these wonderful three-dimensional models that try to create what a person would look like if it was all in proportion. And on occasions, Penfield also was found able to evoke memories at selected cortical sites in awake humans. And if we run forward to the 21st century, then this is Karl Deisseroth, who's a well-known psychiatrist and experimentalist, professor at Stanford, and one of the architects of contemporary optogenetic methods of stimulating the brain using light. So this technique of activating the brain is a way of trying to address some of the causal questions that are not always clear, just straight correlation. But at this point, I need to go to my theme. What is memory? For all of us, it's kind of an act of remembrance. A child being born, a marriage, Christmas celebration, or some other celebration. And I think it's often thought of in relation to sad events. Here's Angela Merkel on the occasion of the 30th anniversary of the fall of the Berlin Wall, lighting a candle at the memorial in Berlin. And we all do these kind of things, and they're important to us personally. So acts of remembrance are one aspect of memory. But of course, with the passage of time, the remembrance changes its character. And there's a kind of transformation from the sadness that Angela Merkel would have had about various friends she would have lost because she, of course, was originally East German. But over time, these expressions of sadness are lost, and what happens is things become expressions of national culture. And this is a picture of the Tower of London on the 100th anniversary of the end of the First World War, and it's the exhibition of the ceramic poppies, one poppy for each person who died in the First World War. But remembrance isn't all there is to memory, of course not. And I think another way to sort of introduce the topic is to think of it as a change in our response to things due to our experience. So a wider sense of memory reflects our capacity, and that of animals also, to change our understanding of the world, our behavior in response to things that happen. And on that view, learning and memory are intertwined, because memory is more than just the folk concept of remembrance. It's a set of specific neural processes through which we represent and encode information into the brain, store and then sometimes consolidate for long-term retention such information. Not always accurately, but nonetheless the process happens. And then express things later through acts of skill or of recognition and recall. So this immediately raises the question of what might be different about the neural mechanisms of memory from that of perception. So let's consider perception first. Neural activity during perception is essentially online. It keeps up with the pace of events. It has to, such as in the visual system. And in acting in real time with the events being perceived or being carried out. So when I show you this picture, initially you may not see much, but gradually a process will happen in your brain such that the Dalmatian dog pops out of the image. And that's kind of perception happening in real time in front of you. But what about the neural mechanisms of memory as distinct from those of perception? Now, the encoding and retrieval of memory involves different types of neural activity from that in perception. And in particular, it somehow manages to mediate a sense of mental time travel, such that you can recall something that happened at an earlier time. You yourself don't move to that earlier time. It's only your sense of things. You have this coexistence with the present time with the past. And this is particularly striking, I think, in the case of music. So I'm going to take the risk of trying to sing something to you. I don't have a very good singing voice, so this is probably going to be a bit of a disaster. I'll try. See one or two notes. Here's, of course, Santuzza singing the Easter hymn in the very famous Sicilian opera, Cavaliera Rusticana. And why it's so special to me is because I first heard it in the Greek amphitheatre Taormina in Sicily. And so as I hear that music, I'm transported back to that evening as the sun set and I saw the twinkling lights on the fishing boats behind me. And so that's a sort of lovely experience for me, but one which clearly involves some element of mental time travel. And so this thing in our heads somehow does all of that. Now, there are lots of numbers, and I know we've got one or two mathematicians in the audience, 100 billion brain cells, 5,000 synaptic connections per cell, gigantic number of connections, and all in 1.4 kilograms. So how does it do it? Well, it's doing lots of different things, motor control, perception, and so on. But with respect to memory networks made up of brain cells, the neural activity somehow represents experience. And the structure and connectivity of the brain changes with such experience. So this is just a global picture of the brain. And nowadays, with modern imaging techniques, we're able actually using, for example, diffusion tensor imaging to see the patterns of connectivity in the brain using non-invasive imaging in the human brain. And it's giving us great insights into how these connections change over time and in the course of disease. So if I go on, let's think about the fact that in perception, neural activity is online. So in the early stage of visual processing, in the so-called lateral geniculate nucleus, cells are responsive to activity in an on-center, off-surround manner. But if you line up 4 such cells in the early stage of visual pathway and connect them together, if they correspond on the retinal bitmap to a line, then at the later, the acortical stage of the visual pathway, you'll have responsiveness that corresponds to a line moving across the screen. And this was the great discovery of David Hubel and Torsten Wiese. So here you've got a combination of certain receptive field properties of individual cells, a particular pattern of anatomy, giving you a result, which might be the first stages of scene processing. Now, this means we really need to clearly add into the picture the anatomical pathways that go from perception to regions mediating higher cognitive function if we're to actually make some step towards understanding things. We can't just do it through psychology alone or through physiology alone. We've got to build in the anatomy. And here, one of the great heroes of neuroscience is Santiago Ramon Picard, who was the early pioneer of using the Golgi technique to look at the connectivity in the brain of lots of different animals. And he gave a cranium lecture to the Royal Society in 1894 about his ideas, which included ideas about memory. Now, of course, things have come on since Cajal, and in one very famous diagram called the Feldman and Van Essen diagram, they map out connections at the time this drawing was made, which was 30 years ago, as they go through these perceptual stages into higher cognitive stages. And it's a very complicated picture, such that people often say everything's connected to everything. And that's not true. But at the same time, there's patterns of connectivity which are the basis of our ability to store knowledge to represent the world. So let's come on to the business of making memories then. Now, the first idea is that there are multiple memory systems. And in particular, the difference between short-term memory, or working memory as we call it, versus long-term memory. And I think we all understand the difference between that. Now, in neural terms, what it means is that the short-term or working memory is continuous neural firing. An event happens, and then there's some continuous neural firing in a circuit which represents that information. And if that continuous neural firing is interrupted, then you lose the information. It's gone. So that's the sort of basis of short-term memory. In contrast, in long-term memory, it seems that there are changes in the strength of connections between brain cells which store the information, and these changes may be structural and biochemical, altering the strength of synaptic connections. And once that's happened, you can then reactivate such memories through partial or associated cues. So let's get you guys to be part of an experiment, and I'm going to make you all part of a short-term memory experiment. So I'm going to give you some numbers and letters and symbols and ask you to imagine that you're in some experiment where you're required to remember this information. So I'll show you the information briefly, and I'll take it off, and you can kind of try and rehearse it in your mind's eye. So here we are. Okay, so what then typically happens in these psychological experiments is that often there are two groups of subjects. One group of subjects is allowed to rehearse the information without interference, and others are given interference. And a common interference is to ask people to count back in threes. So no sooner has that number, those information gone off, you give a number like 57. And they have to out loud say 54, 51, and it's always difficult at the next transition, 48, 45, and so on. And you keep going. And this action of subtracting interferes with this information. And as you'd expect, what's seen on a graph is much poorer working memory results with such interference, showing that this continuous neural activity can be kept in mind, but if you have other things coming in, then you're in trouble. So if we did a test, and I think I'm just asking you to imagine this, you might come up with the fact that you've got two equal signs. Maybe you remember there was a capital letter or two, A and M, might not have got the F, then about the lowercase letters, and so on. So what's often said is there's a magical number, 7 plus or minus 2 is the capacity of short-term memory. And one reason OTPs are only small numbers of things, it's less than this magical number, 7 plus 2. Okay, but I've actually played a little bit of a trick on you, because here they are again. Some of you may feel that you've got it perfectly. I should imagine very few of you. But what I'd actually shown you is Newton's equation, force equals mass times acceleration, and Einstein's equation, E equals mc squared. Now, if I presented the information that way, you'd have all been perfect, right? Many of you are scientists, would have no difficulty with that. But because I scrambled it and didn't give you that prior knowledge, I was making it hard for you, as it were. And this leads to a radical discovery, which is that by virtue of all these pathways that Feldman and Van Esten discovered, information from central systems actually accesses factual memory prior to going into short-term memory. So the astonishing thing is that long-term memory is actually the input to short-term memory, not, as many people suppose, the other way around, which I think is quite a radical kind of idea. Now, the second thing is the fact that when we go into long-term memory, there's a huge division between the learning of skills of various kinds, whether it's tennis, golf, or riding a bicycle, or what have you, and what's sometimes called declarative memory. Gilbert's Ryle's learning how versus learning that. And procedural learning goes through actions and then becomes habitual, whereas declarative memory gets divided into further categories, which I'll come on to. So what we now know is that these two big categories of long-term memory are mediated by radically different areas of the brain. And in particular, the procedural learning system is mediated by the striatum, and the declarative memory system by the hippocampus and cortex. And our key bits of evidence for this come from the very careful study of patients by neurologists and neuropsychologists, who have shown that in amnesic patients, you get huge deficits in declarative memory, but not necessarily any deficit in procedural. And from the study of people with either damage to the striatum or damage in particular to the dopaminergic input to the striatum. And that's shown here, where in the rodent, for example, there is lots of projections going up from the striatum and into the cortex. And this acts as a kind of reward signal for the learning of particular skills. So, what of this big difference? Well, it was first discovered in the context of amnesia by Brenda Milner, neuropsychologist at McGill University in Canada, who was studying a patient called HM, and she trained HM to do mirror drawing. He got really quite good at it. But when she brought HM into the testing room after a few days, she said to him, let's try this mirror drawing. And he, being a deeply amnesic patient, said, what mirror drawing? I can't remember. But she said, well, I know you can't remember, give it a go. And then he did it perfect. So you get this astonishing dissociation between the skills and the declarative memory. And I'd just like to illustrate this to you in relation not to an amnesic patient, but in relation to Alzheimer's disease. Because Brenda Milner always saw this as a sort of one of the kind of magical moments in her career, this discovery of this dissociation without any awareness. The text is in Spanish, but it doesn't matter.
Some of them were going to we watched them play. White starts as bright as the men still lying back.
I think it's a very striking example of that association and quite touching. So let's come to declarative memory, which is what I'm going to devote the rest of the talk to thinking about, episodic, semantic, and spatial. Now, episodic memory has to do with events. And the thing about events is, in many cases, they happen just once, like the child or something like that. So I've been trying to think of what might be a suitable event to refer to here in India. I don't know, I may get this wrong, but let's try and see if this is one that the older people know here. The shot of hope that turned the World Cup final of 1983 in India's favor. Many of you may remember that. Of course, it's not two shots, it's just one shot. And so I'm imagining that Srikanath remembered that moment for the rest of his life. So the sense that there's just sort of one moment was captured by the French photographer Henri Cartier-Bresson. She called the decisive moment. There's particular moments that you kind of capture, and that's really what the episodic memory system do. So unlike the procedural one, which you have lots and lots of trials to get better and better at riding a bike or whatever this particular skill might be, mirror drawing in the case of HM, you have just a single event, and the plasticity system has to be able to cope with that. Now, with respect to semantic memory, it's our factual knowledge of the world, and it's built from our experience of events and so on. But we strip the context and spatial and temporal tags from that and build up systems of knowledge, which may well be hierarchically organized or perhaps more disorganized, depending on the particular person. So in Britain, we often sort of say that the archetypal example of this is do you remember when you first learned that canaries are yellow? And of course, none of us do. We all know that canaries are yellow, but we don't remember the moment that actually happened. And then spatial memory. Here's a map of Edinburgh where I live, and I know this city reasonably well. Now, the structure which mediates this is the hippocampus, which sits within the medial temporal lobe of the human brain, and it's also present in all mammals, in fact, in many vertebrates too. And if you look at crescent violet sections, you can see that the structure in the human brain and the structure in the monkey brain and the rat brain have enormous similarities. Now, if you use Cahal's technique of the Golgi, you can actually see the individual cells and how they're laid out. You've got a cell body layer here and the dendrites coming down, and another cell body layer here, and so on. Now, a dramatic experiment was done just about 40 years ago by an American scientist working in London called John O'Keefe. and he discovered what's called place cells. He lowered an electrode into the hippocampus of rats that were moving around. He did it in an earlier surgical operation and then adjusted it to be right beside the cell bodies. And what he found was cells that responded to the particular position of the animal in space. So if this is his recording electrode, it's being lowered down into the hippocampus. And then as the animal moves around, nothing much is happening, and then suddenly cell fires like that, and then stops firing. And then as the animal comes back, at exactly the same spot in space. And he did all sorts of controls to check that it wasn't due to particular sensory cues. It's to do with a position in space. So the hippocampus seems to sort of know where we are, as it were, and represent where we are. And that discovery was followed by others, the so-called head direction cells, which are cells, again, I'll try to imitate. If I'm recording from a head direction cell, which fires over there, then nothing's much happening, then it goes. What matters is what direction I'm pointing, not where I am, right? And then finally is the grid cells, which were discovered by Edvard and Margaret Moser. And they're a kind of metric of space. They're like place cells, they fire in particular positions, but they fire repeatedly. Like that. And they're laid out in a wonderful kind of triangular pattern. And they're all in adjacent areas of the brain, in the hippocampus or around the hippocampus. So I was inspired by John O'Keefe's discovery of place cells and had met him in London. I then got my first job at the University of St Andrews, and he encouraged me to try to develop some kind of behavioral task that might capture the notion of finding a point in space without the use of any sensory cues to guide you there. And here I had a stroke of luck, because I was appointed as a lecturer, but to a department that had no lab facilities. So I did my teaching for a year, and I went to the head of department, Professor Malcolm Jeeves, and I said, it's great, I'm enjoying it here, but I really would like to do some research. And he said, leave it with me, I'll find you a place. So what he did was he asked around, and eventually space was found in this inauspicious building, the Gatti Marine Laboratory of St Andrews. And I was given a couple of rooms there. And every day I walked past all sorts of sea creatures at the entrance to this place. And that was lucky, because then I thought, gosh, I wonder if rats could swim around in water, and that might address my problem that I need to do. And in particular, I thought, well, if I devise a big circular swimming pool where there's a place of safety at one particular point in space there, the animals will never see it, they'll never hear it, they won't feel it until they get to it. And by swirling up the water, there won't be any smell. And so that's what I did, and I developed an apparatus called the water maze, which has been used by my lab and others for some while. So just to give you a taste of how this works. So there's the animal finding the platform. He's been extensively trained. It's held down by electromagnets on a spindle, and now the animal's put back in. This is the first time he has what's called a probe trial. And what I hope he will persuade you is that he's acquired knowledge of where in this pool it might be safe. There's no platform. He's having to execute behavior very different from what he would do on a regular training trial. He's engaging in some sort of searching behavior. But you can see, as we track online, it's relatively accurate. And we use this task to establish that if you damage the hippocampus, the animals couldn't do the task. And then later, in experiments on synaptic plasticity, if we blocked synaptic plasticity, they also had trouble in learning. Okay. But there's a problem. And I want to share with you something that's related to some very recent research we've been doing. And the problem is as follows. I've explained to you about place cells, And place cells define where I am. This place cell corresponds to this place. Another place cell will correspond to this place. Another place cell to that place, and so on. So if place cells are defined as cells that fire if and only if you're in that place, that's fine. But how do we then use them for navigation? So if you've got an animal in place A, there may be 3 place cells firing there. But if you want to go to place B and not to place C, you somehow from A need to activate the information about place B and activate the information about place C and make some decision about where to go. But by their very definition, you can't because you're not in that place. So that kind of logical problem has hung around the field for some while. So we decided to explore this using a new technique called calcium imaging in a different apparatus than the water maze, because we wanted physiology and water mazes don't go very well together. So here's an animal coming out into an arena where there are sand wells. He finds the right sand well, he digs up some food, and like any self-respecting rat, he takes it back home again. So what we did was we used a technique that was developed at Stanford University, in which you have miniature endoscopes, which weigh only 2 grams. They have a CMOS camera in them, and a gradient refractive index lens is passed down into the brain, very narrow diameter, and down to the hippocampus. And what you see then is if you've virally expressed something called GCAMP6F in the hippocampus, you can see calcium transients across all these cells. And this diagram shows you very nicely these calcium transients actually happening. Here's one particular region of interest, another, and so on. So, what's the experiment? Well, you can see where in a particular part of the hippocampus we've actually expressed our GCAMP6F. Here's the camera on the rat. Here's the grin lens coming down to near the hippocampus. Your initial raw data looks a bit like this, but if you compute the delta over F, then you can see the various regions of interest popping out as the cells fire. And in an experiment led by Francesco Gobbo in my lab with 5 animals, we managed to get about 190 cells in area CA1 in each animal. So we then train the animals in this task, in which the first phase is looking for the place cells, then there's a sample phase in which they have to find the correct platform, the correct sandwell. And that correct sandwell changes from day to day. So each day they've got to form a recent memory. And then they have a choice trial. And what you can see is the animals get really quite good at this task, such that they're forming up at about 80% correct. And if you look also at the paths, on session 2, they're a bit all over the place. Session 9, they're getting better, but by session 19, they're able to remember where to go. Now, the critical bit of the experiment comes next. What I'm interested in is the animal making a decision about where to go on the basis of information which, by the traditional definition of place cells, would be unavailable to him because they'd be corresponding to somewhere else. So what we did was we recorded the animals in the start box in this arena. Saw the animal a moment ago in the arena. And what we saw was that the rate of events started to increase over the 10 seconds prior to the door opening and the animal coming out. And then we created a decoder with respect to what these events were. And it turned out that the decoder showed that the animal was anticipating correctly where he was going to go. And if you did a shuffle, the chance level was about 33%. And so the data shows that on correct trials, they in the start box are already starting, now that we're recording from enough cells rather than just odd individual ones. There's a sort of population of cells that corresponds to going to place B and not to place C. And you can see that in this population recording. So here we have the animal in the start box. You can see these are probably place cells from the start box. But even though he's sitting there, he's starting to see some cells that correspond to where he wants to go to. And the same thing's happening here. He's perhaps imagining the destination. That goes on. And then he has second thoughts. And so there's a brief moment at -6.8 seconds where he thinks he should go over there, but then he changes his mind again and back to the correct spot. So we can watch the sort of dynamics of this decision making happening in front of us. Now, because we've got population recording, we can use what's called manifolds to reveal patterns that correspond to the movement of the center of gravity or the centroid of these activated regions of interest as the animal move around. So at the beginning, they're shown in blue, and they get to the end, they're shown in red. And what's quite nice is that these are all in circles, such that the red bit and the dark blue bit correspond to the same location here in this two-dimensional space. So by using this population recording, we can sort of unmask what's actually happening from these manifolds. And that happens on the sample trials, choice trials also, at the end point and the start point are at common positions in space. So that's an example of representation. But I think it's also an example of, at least over a very short period of time, mental time travel. The animals here is imagining what might have happened on a previous trial, is imagining maybe what he's about to do. And we're seeing that in the end. Now let's come to keeping events. Now, the study of forgetting has a long history. It goes back to the German experimental psychologist Hermann Ebbinghaus, who did an original studies just on himself learning nonsense syllables. And he found that at 24 hours, there was only about 33% retention. That suggests there's dramatic loss of information during the day. And I guess that's the experience that most of us have. But is this information lost randomly, or might there be some selectivity about how it's happening? We remember what we attend to, sure. Remember what we're interested in, sure. We remember well when we're not under stress, a topic that's been of great interest to Shona. And so from the stream of attended events of the day, some things are remembered, but not all. Now, people doing non-invasive functional magnetic resonance imaging in humans have tried to look at this by using a technique called event-related functional imaging. And if you do that and classify information that you test later as being remembered, later as being familiar, or later as being forgotten, you can see that the signal you get at the moment the information is given is largest for the information that's remembered. And that's shown in a number of regions, including the parahippocampal cortex, this particular task. So that points the finger at things that are happening at the moment of encoding. But I don't think that's the whole story. In fact, I very much don't think it's the whole story. I think there are things happening at the moment of encoding, but there are things happening beforehand which will influence how well you remember, and there are things happening afterwards which will influence how you. And a clue to thinking about this came from the discovery of the phenomenon of long-term potentiation in hippocampus by Timothy Bliss and Tarrier Levin. And this is a classic paper from the journal Physiology, in which they recorded various potentials, and then they gave brief bursts of high-frequency stimulation, and they found the potentials got bigger on the pathway on which they'd done that, but on the control pathway things stayed stable. And this potentiation of synaptic connectivity became the bedrock of a lot of work trying to understand exactly how this happens. And so the idea has developed over many years and many, many experiments that this is a process through which the strength of synaptic connectivity between neurons and hippocampus can be enhanced by specific patterns of neural stimulation. High-frequency stimulation is one, but there are many others. Interesting, it uses an automatic Hebbian rule for synaptic change, not an error-driven. So unlike the striatum, which is constantly trying to correct your skills as you develop them, this is kind of capturing everything with an unsupervised learning algorithm. And we're beginning to understand the basis by which LTP is mediated in terms of the individual glutamate receptors that are expressed on the postsynaptic side of these synapses. I won't go into that in great detail, but just to mention that the second big discovery here was made by Graham Collingridge, who found that a drug called AP5, which was later found to be an NMDA receptor antagonist, a particular type of glutamate receptor antagonist, would block the induction of LTP Once you washed it out, you could get your LTP. A major discovery which has been replicated by many others since. Now, at this point, the molecular biologists began to think very carefully about the structure of NMDA receptors. Of course, the structural biologists got involved as well. We now have a pretty detailed understanding of the way the NMDA receptor operates. In particular, the ion channel associated with the receptor is blocked by magnesium, and such that when glutamate binds at the glutamate binding site, ordinarily nothing happens. However, the magnesium is released if the postsynaptic side becomes depolarized. So if the postsynaptic side becomes depolarized, or if the postsynaptic side has a shell firing, magnesium is released, and then glutamate binding to its binding site will allow calcium to enter through the ionophore. So basically, this is a biological AND gate. It requires the conjunction of two things. On the presynaptic side, the release of transmitter. On the postsynaptic side, depolarization. And we're now beginning to really understand this in terms of the underlying structural biology. So what we have is a system in which we can increase from a basal state the number of receptors, that's the potentiation, or we can decrease them, and that's the depression. Well, that's all very well, but how far does that get us? Well, here I think we have to be careful, because many people suppose that we need to understand this only at the synaptic level, but I would argue, again with reference to my claims about anatomy, that we need to think about the anatomy as well. Suppose we embed this potentiation mechanism into a simple reflex. So you're going to have LTP induction at a particular point in a circuit which goes from cell A to cell B to cell C. What will happen in such a circuit is that if a particular input normally gives a certain output, once you induce LTP here, you'll get a bigger output. So you'll have potentiated the synapse, and that will give you a bigger output. And many people suppose that's the basis of memory, but it's not enough. You also need to think about the particular type of circuit in which this plasticity is embedded. And if you have what's called a distributed associative network, here you again have cell A, B, and C, but a particular Q might come in here, and by virtue of the firing of these cells, you'll get a pattern of potentiation that depends upon this conjunction, depends on this AND gate. And if Q2 is involved in firing of these cells, you'll develop an association between Q1 and Q2 that depends upon the conjoint properties of potentiation on the one hand and the anatomical arrangement on the other. You need both of those things. So just to sort of illustrate this, and this is a sort of Mickey Mouse way of doing it, if you imagine a binary number 0011, and that's not unrealistic. You've got neural spikes are active or not active, so that's okay. And you combine that with another binary number 1010, then if you operate this rule, the NMDA receptor rule, sometimes called the Hebb rule, then the potentiation will happen at the point where you've got presynaptic activity of 1 here and postsynaptic activity of 1 there. 0 and 1 is not good enough. 1 and 0 is not good enough. 0, 0 is not good enough. But you have to have the 1 and the 1. So that will give you, in this simple 4x4 matrix, 4 places where synaptic potentiation has occurred. Now, if you now leave the matrix in that state and add in this one now being associated with something else, 1010, it's this one being associated with 00110, then you'll add in 3 more. And if you imagine this with 5,000 synaptic connections on each cell, you can see you've got a fantastic potential storage capacity if this really does work as a memory. So let's see what happens when we give the memory in its final state 0011. So what will happen is the current will go in, it'll whiz around at this potentiated synapse, and this one, and give us an output 2120. Now 2120 is obviously different from 1010. But what you have to do then is integer division. And you have to divide by the number of active elements, 1 and 1 here. And if you divide interdivision, forget the remainder, you get 1010, which is this one. So the system works. Now you might think the interdivision is a bit of a sort of cheat, but actually, if you look in the hippocampus, there are lots of inhibitory neurons which project onto the somatic cells, and they don't show LTP. So the probability that they will fire will be proportional to the number of active elements. And so perhaps the inhibition is performing the mathematical operation of interdivision. So when we then look at the structure of the hippocampus, we've got various areas, dentate gyrus, CA3, CA1, and so on. But notice how strikingly similar this structure is to these simple unsupervised distributed dissociative memories. I think it's a great discovery. This is not my discovery at all, but I think it's a great discovery. So that means we can store information in this. But there's another important twist to the story here. Because there are at least two and possibly three distinct forms of long-term potentiation. LTP1, LTP2, and LTP3. LTP1 decays to baseline within about 3 hours. So it forgets. And perhaps we all think naively forgetting's bad. I want to argue forgetting's really good. But LTP2 lasts for a much longer period of time. And it involves translation of RNA, so it depends upon protein synthesis. And then you can get something lasting at least 24 hours, perhaps longer. And then other mechanisms will kick in to keep that information. So what we've got is a situation in which if you plot memory trace length on a y-axis, it goes up rapidly at the encoding point, just like I've shown you in the distributed associative matrix, stored for a while, and then if nothing else happens, the memory will go. But if during this period you get dendritic mRNA translation, the mRNAs are out in the dendrites of the cells, in the synthesis of this of plasticity-related proteins, structural changes will happen to spines, and that may enable you to keep that information such that later on, when you have retrieval and successfully retrieved, rather than forget. Now, some years ago, I published a paper which addressed the puzzle about where the plasticity-related proteins would go. Because they may be diffusing randomly, but how do they get captured at the synapses which have been potentiated? And so we did a series of in vitro brain slice physiology experiments, some done in Germany by Uwe Frey, some done in Edinburgh in my own group, in which we had three stimulus pathways, which are shown here, one recording electrode. And we were able from that to actually record excitatory postsynaptic potentials, which go negative in extracellular recording. And you can see that that's happening, and we can measure the slope of this line both before and after LTP. So here you have strong tetanization. And that strong tetanization gives you an LTP, which then decays back down to some elevated baseline. And if you plot the data from 2 hours to 10 hours, you can see that it's relatively stable. Now, suppose instead you induce LTP with a very weak stimulus. That weak stimulus is unable to trigger RNA translation, unable to synthesize plasticity-related proteins. So what happens is you get potentiation. It's not too bad, 40%. But if you track it compared to the unpotentiated pathway, it decays to baseline after about 6 to 8 hours, maybe less. Okay, so we've got potentiation that can decay, potentiation which is stimulated so strongly that it lasts. What happens now if we put those two things together? Well, what happens is that something magic happens, which is that the weak pathway now fails to decay. So we've got some strong event happening, and then maybe 40 minutes later or 40 minutes before, the weak one, which normally decays to baseline, now lasts for a much longer period of time. So what might be going on? Well, these set of rather sort of Mickey Mouse cartoons try to explain the idea that at the point that you set long-term potentiation, you also set a tag, which in America is called a baseball glove, but in India is called a wicketkeeper's glove, right? So the wicketkeeper's glove is going to capture plasticity-related proteins, and when this capturing happens, it will stabilize the synaptic change. Now, if you have weak stimulation, you set a tag, but you don't induce this translation of the mRNAs to give you the plasticity-related proteins. But if you put the two things together, then what happens is the weak stimulation will give you late LTP because it hijacks the other guy's plasticity proteins within the window of time that's permitted, right? So synaptic tags set by the weak stimulation hijack the plasticity proteins induced in response to the strong stimulation or that of neuromodulatory pathways, and so enable late LTP. And we also did experiments using RNA translational inhibitors like anisomycin and got basically the same kind of result. And we've gone on and done further in vitro slice experiments to investigate this in more detail. But what I now need to do is to take you into the behavioral experiments. So we then did some behavioral experiments in which we go back to this little event arena that I showed you before in the film. And during an encoding trial, the animal has to remember that today, Monday, the food is to be found in row 2, column 2. And then after either 30 minutes or 24 hours later, we test, can the animal remember that? Well, the answer is that he can. So if you test him after 30 minutes, he remembers perfectly well. He's spending 50% of his time at the correct spot. But if you test after 24 hours, the memory's gone. So this is the weak memory decaying over time, now shown in the behavioral domain. But this is the data I showed you earlier from the LTP. So that's great. And I'm struck by how similar the bar graphs are for the LTP data and for the behavioral data. So how do we mimic in this behavioral experiment a strong stimulation? Well, the way we do that is to give the animals a very novel experience. And we do that by putting them into a plastic box for 5 minutes or so, either just before an encoding trial or better, just after. And we know from a lot of experiments that other people have done that this activates a variety of immediate early genes and also triggers RNA translation. So this process itself will activate the availability, will trigger the availability of these plasticity-related proteins. So now we just simply change our experimental design. It's a different day, so we're in row 2, column 6. We then have this novel experience happening 30 minutes after encoding, and the novel experience does not provide information about where to go. All it does is upregulate the availability of plasticity proteins. That's all. But then when we test it here 24 hours later, now they can remember. So if you have just one pellet encoding, which normally gives you forgetting after 24 hours, if you combine it with this novelty 30 minutes later, you get perfect instant. Now, time's running on, but if I may, I'm going to keep going for a little bit if I can. So we then went on to do optogenetic experiments because we wanted to find out what the source of this novelty signal is. And to cut a long story short, I won't take you through this in any great detail. We provided evidence that it comes from the noradrenergic system. And we did that by expressing channel rhodopsin with the yellow fluorescent protein into the LC, also to BTA. Here you see co-localization of the tyrosine hydroxylase in these PH-CRE mice together with the channel rhodopsin signal. And then we devised a particular experiment in which now, instead of giving them a novel experience, we put them in their home cage and just simply activated using optogenetics these set of cells in locus cerebris. We tried it in BTA, it doesn't work in BTA, but it does work in LC. So now we're going to have our encoding, then we're going to fire these cells 30 minutes later, and then ask what happens 24 hours later. And you'll be pleased to see that indeed we do get perfectly good memory if that happens. So we've sort of identified, we think, the cells that can at least mimic this novelty effect. Whether it's the only group of cells, we don't know, but it seems that they can. There's some other pharmacological oddities to this data, but I think I won't dwell on those. So what I presented to you basically is a system which uses an unsupervised learning algorithm, a Hebbian NMDA receptor basic mechanism, which most of the time gives you forgetting over a period of about 3 to 4 to 5 hours. But if at some point of the day something happens, which is novel and exciting, you upregulate these plasticity proteins, and that memory will get kept. So it's not an error correcting algorithm, but it is a selectivity. And that's happening through these tags, their interaction with the plasticity proteins when the things overlap. Now, why might that matter? Well, it's a very ingenious system that evolution's come up with here, which is that you capture everything online. You don't have to make a decision at the moment encoding happens, whether this is important or not. You just capture everything. But you've got some window of time, maybe of a couple of hours, to determine whether this is worth keeping or not. We've used novelty, but it may be prior knowledge, it may be whether it links into your professional knowledge or what have you. All sorts of ways in which the selectivity might operate. And I think strikingly in the case of flashbulb memories, the certain events are very well remembered by the particular generation and geographical locale for which they're surprising. My mum never stopped telling me about how happy she was on VE Day in 1945. I'm just old enough to remember the assassination of President Kennedy. I know exactly where I was when I heard that. I was in Sacramento for Princess Diana's death. 9/11, I was at Stansted Airport, and so on. And perhaps in your own culture, there'll be similar kinds of events. And what I'm trying to put to you is that you don't just remember those horrific images of Twin Towers burning, but a distinctive feature of these memories is that we also have memory for many of the trivial events of that day, who we were with, whether we were having a cup of coffee, where, and so on. And I tried to offer you an explanation of why that might be. Now, let me just, in the last 10 minutes or so, turn to the losing of memory. I've already mentioned to you that most of us think of forgetting as unfortunate, and of course, in many instances, it can be, like when you tell a joke back to somebody who just told it to you the day before, or something like that. That kind of thing can be very embarrassing. But I've also tried to, in the last section, explain to you why forgetting so valuable, because it builds into the system the opportunity to capture lots of things without deciding whether you want to keep them or not. It stops the memory system from getting saturated and gives you a window of time to decide. Now, somebody who developed a similar kind of argument was Dan Schachter, who talked about the 7 sins of memory. These are failures of memory that are certainly exacerbated by age. But Schachter argues that not really failures of memory at all. They reflect the proper operation of a finely tuned system. He writes, the 7 sins are byproducts of otherwise adaptive features of memory, a price we pay for processes and functions that ordinarily serve us well. So we can think of this as forgetting as a kind of intelligent error. Let's take the sin of persistence. Clearly, post-traumatic stress disorder, PTSD, can be very, very disabling. And I recognize that. But if you think about it from the point of view of evolution, persistent memory of successful escape from any life-threatening situations is adaptive. So there's 2 sides to the thing. Sin of transience, forgetting things quickly is annoying. You know, where I put my glasses this morning, that kind of thing. But imagine what it would be like if we failed to forget most of the events of daily life. So don't think of all forgetting as bad. Some of it's quite good. It's a property, particularly of your episodic memory system and value. Now, another contribution to forgetting that I know many people are worried about, particularly parents of young children, is can the excessive use of digital devices undermine? And we could probably argue about this over a beer till cows come home, or whatever they do in India. Cows do other things, I guess. But is cognitive function being affected by too much social media? The extensive use of digital devices? Well, as you might imagine, some people are trying to look at this experimentally. And so far, the picture from the few experimental studies, the few good experimental studies that we've done, is actually rather mixed. Many studies report clear impairments from the extensive use of digital media, particularly in people's ability to find their way about as they become more and more reliant on GPS. But other studies have reported enhancement. So the picture at the moment is we just don't know, but it's a sort of watch the space situation. However, clearly forgetting can become more serious, and notably in the case of Alzheimer's disease. It's the most common form of dementia. It affects a staggering 55 million people worldwide. It's everywhere. It's extremely expensive in terms of the burden, healthcare, particularly the carers at home. So far, there's no cure. It's beginning to be treatments, but nothing very much. And in many respects, it's a disease that affects the carers as much as the victim, empathetically revealed in John Bailey's account of his life in Paris. Now, it's characterized by various stages: a sort of forgetful early stage, which then gives rise to confusion, severe dementia, and finally a terminal stage where there's almost complete loss of intelligence. Now, the biochemical markers of this include the famous amyloid plaques and tangles that can be seen in stained tissue in neuropathological specimens taken from patients who suffered and died from Alzheimer's disease. And it's tempting to suppose that those are the cause of the disease. And if we could somehow stop this amyloid beta from building up, or we could stop the phosphorylation of tau, perhaps we would then have a kind of a cure. But the problem is we've got a sort of chicken and egg problem. For the case of familial disorders, there's say a gene defect, and that may result in the accumulation of a beta peptide. But is that really causing, or is that some kind of protective mechanism? We just don't know. It could be either. And does that lead to neuronal injury? Or do you get neuronal dysfunction first and then pathological changes later, which seems more likely? And at what point in this equation does altered power metabolism? And there's lots of questions here about the secrets of things, and it's a very active and a very important area of research to try to work this all out. But I should like, somewhat self-indulgently, to touch on a personal foray into animal models of Alzheimer's disease, which I had an opportunity to get involved in, following the publication by Elan Pharmaceuticals in 1995 in Nature of the PDAPP mouse. This was a mouse that overexpressed human mini gene of APP with the 607 codon mutation. And what they published was that this mouse developed amyloid plaques in a nice age-dependent way. And I wrote to the company, actually I sent them a fax, it's that long ago, saying, are your mice forgetful? And I was delighted when they wrote back saying, would you like to find out? And so I began a collaboration with Elan. I never took any money from them, but I was thrilled to have access to their animals. And we started to do some experiments to see, did these animals have a learning deficit? And indeed, we found they did. And I think the next graph shows very clearly that if you looked at these animals when they're young, middle-aged or old, and old is 22 months old, then the PDAPP animals are forgetting faster than the non-transgenic aged animals. And this we showed in both the cross-sectional study and in the longitudinal study. The longitudinal study was probably the longest experiment they ever did. It began with the animals when they were very young, and the experiment ended when they were 22 months of age. But it was a very clear result. And then Elan published, 1999, a paper from Dale Schenken's colleagues, an antibody 3D6, which would clear plaques in the brain. Striking finding. And the pictures in his paper in Nature 1999 show a pediatric animal full of plaques, and then a pediatric animal who's had 3D6 and the plaques are gone. So that was a kind of a very exciting moment to try to address this causal issue. So a few years later, we were able to do a study with them in which we looked at immunization of our PDAPP animals using the various tasks that we've been looking at here. And we tried the immunization against this excessive beta amyloid that you had in these transgenic mice. And we had only partial success in rescuing the memory deficit. But that was for two reasons, one of which is uninteresting and one of which is, I think, interesting. The uninteresting one is that in these transgenic animals, there's about a 7 times overexpression of the human mini-gene. So even if you bring it down by 50%, which is colossal, you still at 3 times overexpression. And maybe that will do nothing, right? So it's the problem with these transgenic animals. They can tell you a certain amount, but they're not perfect. There are problems here. They're basically kind of furry test tubes in which we can do whatever experiments we can do, but we have to be careful about these model animals as to what we can learn. But there was a second reason why our results, I think, may be of interest. And that is that we did the experiment 2 ways. We immunized some animals from a very young age. We knew we were dealing with a familial disorder. We knew these animals were going to develop plaques. So we started treating them before they developed plaques. And in the second part of the experiment, we waited till they were 11 months of age. And the results were clear-cut between these two, that the only animals in which we could rescue cognitive function were the ones in which we gave the immunization early enough. If we gave it much later, we couldn't bring the A-beta levels down, and we couldn't see a rescue of cognitive function. But you can see there's a small number of them in which we were successful getting comparable performance in our learning capacity tasks. This is for hippocampal total A-beta, this is cortical total A-beta, and these animals are performing very well. So why does that matter? Well, what you're seeing here is PET imaging of amyloid plaques in humans. And notice at this point, this is 14 years before diagnosis, 10 years before diagnosis, 7 years before diagnosis. So just as in Parkinson's disease, these changes are insidiously happening in the brain long before the people start to show refractive deficits. And this is a fundamental problem in the field. And it's a problem really for the pharmaceutical industry too, because they're only allowed to test their drugs on people who've already been time-based, which, if we dare extrapolate from my Mickey Mouse experiment, then is sort of the wrong thing to do. You need to capture them early and be able to see if you could alter the progression of this. However, there has been a breakthrough recently with a drug called aducanumab, and another one which is even better called lecanemab, in which they're looking at this in the very earliest stages of Alzheimer's disease and seeing both a reduction in plaques and also some decline in the rate of decline of cognitive function. So there's hope. So I've taken far more of your time than I should. But thank you. So my concluding thoughts really are that we need to think of memory not just as an act of remembrance, but as a kind of family of processes through which we encode information, we keep information, and we sometimes lose information. And I tried to say a little bit about the brain regions and activity patterns that mediate the encoding of information into memory, particularly the hippocampus, for declarative memory and striatum skills. Something of the neural mechanisms, such as synaptic plasticity, that underlie the storage and consolidation and the selectivity of memory retention. And then a few words just now about forgetting. But what thoughts to leave you with? Well, memory defines our individuality. You know, yes, we have our genetics too. Of course, that's a big factor, but what I remember, what Shona remembers, overlaps with existing. Same for all of you. And I think that shared memories within families and amongst friends acts as a kind of glue that holds us all together. I'm sure you can think of examples in your own family life of how important those memories are. So, with respect to memory, cherish it. Thank you very much.
Thank you very much. That's a memorable. We'll take questions. I need questions, not comments, preferably questions, and preferably from the other.
That was a wonderful talk. So I have two questions. First, I might have missed it when you showed the place cells and in the place prediction cells. Are they overlapping populations or are they distinct populations mutually exclusive from each other?
They're overlapping. I think many of the cells that we record in the start box are place cells out in the environment. But the key difference in this task is that ordinarily place cells are recorded by just letting animals wander around an environment with nothing much to do. What we've done is we've built in a task where they have to use that information and also are able to record for as many as 200 cells simultaneously. So the combination of those two things makes it possible to see this element of prediction that had not been seen previously in just straight exploration.
So how do you think it could separate the place it is currently in versus what it is thinking of going to in terms of population coding?
From the calcium imaging, it's rather difficult to do that because all we're getting is activity in a region of interest. But it might be possible with more elegant multiple signal unit recording techniques if you could get enough cells to sort of mark the individual neurons and see if they are a different subset of cells. That might be possible. I think that would be quite difficult to do just with calcium imaging.
Thanks. And my second question is about the Roman method of remembering things. The Roman method of remembering things, where they imagine a palace and put things in the palace around. So this association makes it much easier to remember facts.
Right.
And how do you think that works? Is it a separate kind of memory where you, or is it some kind of compression of information?
Well, what a very interesting question, referring to very famous book by Francis Yates, The Cultural Institute, about the art of memory. It uses these techniques of memory theatres and so on. So, from John O'Keefe's point of view, he sees place cells as part of a navigation system, and he's interested in how animals find their way around. I prefer to see that as one aspect. The wider aspect is to do with the fact that in episodic memory, we actually automatically bind our memory of events with the places that we're at. So the fact that the hippocampus is a spatial system, but it's also doing episodic memory, is exactly as one would expect. You've got the opportunity through this distributed associative memory system of binding events to context, binding events to places, and that's going to be happening automatically. So to try to sort of illustrate this in an anecdotal kind of way, let's suppose on your way home tonight you see a car accident. Yeah, hope you don't. Let's suppose you do. When you get home and you're with your family, you say, well, I saw this car accident on the way home. What might they then ask? They might say, well, was anybody hurt? That might be the first question. But then they might say, where did it happen? Suppose you said, well, I can't remember where it happened, but I know I saw an accident. That would be strange, right? You sort of automatically bind the event of the accident to where it happens. And I think the fact that those two properties are part of the hippocampus, this spatial system, plus this event memory system, is no coincidence.
I have also two questions. Question number one. As you told that in the medicine and research, experimental Okapi and Latron other neuroscientists they established the navigation, the spatiotemporal information that gets encoded in the population pool, which occurs in evokes in the population, red cells and red cells. Now, my question is that the information, spatiotemporal information getting encoded in the population pool. Is there any higher order neural fix which interpret this encoded information in the mid-temporal or in the higher level of the brain? This is my first part of the question.
Right. So, of course, the hippocampus does not operate in isolation. It's getting inputs from lots of different areas. And there's the ventral visual pathway, which is providing information into the parahippocampal cortex and then through the entorhinal cortex of the hippocampus. But there are also more dorsal projections from the retrosphenial cortex. And one of the issues that people are facing at the moment is how the transformation happens between egocentric and allocentric frames of reference. Because the initial frames of reference are entirely egocentric, something to the left, something to the right. But the way the hippocampus operates is forming allocentric kind of maps of space. So there's that transformation going on. But a further facet of this is the opportunity for additional kinds of information, such as temporal information, which you're alluding to, which could be represented in hippocampus and may be very important for events. Now, Howard Eichenbaum, sadly is no longer with us, discovered time cells, and he was able to record from cells that happened fired at particular points in time in some moment in time, suggesting that temporal information is available there. Now, the last thing I want to sort of say is that if you look at the anatomy, and I gave you an example with distributed associative coding, what I didn't do is explain more about area CA3. So in area CA3, you get a feedback onto itself. So the axons of CA3 neurons feed back into other CA3 neurons. Now, this may be very important in event memory, because in event memory, you need to know that event A happened first, then event B, and event C to get the sequence right. If you don't get the sequence right, it will go wrong. And so some people are exploring the idea that CA3 gives you this sequential information. So maybe that's getting at the kind of things that we're thinking of.
I'm more pinpointing my question.
Right.
That the population code, whenever we study, we assume that it has reached the stabilized condition and the spatiotemporal information are encoded. We model it normally in the modeling point of view.
Right.
We model the population code by some probability distributed function or whatever.
Right.
And suppose I eliminate the temporal part, but spatial part, the coordinate of the point source may be encoded as a mean of the population code.
Right.
Let us start. Now, the population code has a transient time also when it grows.
Sure.
So, can I model that there is a higher order fix of neuronal architecture which parallelly starts estimating this mean of this population code? It executes during the time and evolution, it executes certain trajectory in this case. And there is a higher order neuronal fix, which again starts estimating these things, and where they intersect, that is the point. They assume this is the coordinate of the.
Well, I honestly don't know the answer to that. I think we're pinpointing a very interesting issue. But I don't think I can answer that one specifically, because it would depend a little bit on the nature of these models, but we could talk about it. Yeah.
Thank you, sir, for the wonderful talk. Regarding the two forms of scientific plasticity, long-term potentiation and long-term depression, could you tell us a bit more how LTD or long-term depression is related to forgetting or losing memory?
Yes, so there has been work on this because if potentiation is the incorporation of more AMPA receptors into the postsynaptic density, and that's the sort of structural basis of the potentiation, then you expect LTD to produce some kind of forgetting. And there have been experiments which have investigated that and found that to be the case. The problem I think we've got at the moment is we don't really understand the ordinary normal circumstances in which LTD happens. Because the way it's done in physiological experiments is often long trains of 1 Hz stimulation, which are very unphysiological. Now, there's been some suggestion from spike time independent plasticity that maybe precise timing of things can produce LTD, and that may be more realistic. But there haven't yet been in vivo experiments to really examine whether that's the case. So you can produce, in vitro, you can produce long-term depression by these long trains. It's very difficult to use those in vivo. But what you can do is examine the impact of drugs that either promote or slow down AMPA receptor endocytosis. Those have corresponding effects on forgetting of a predictable kind. So it looks like the idea is on the right track, but we're not yet there yet.
It's a wonderful talk. Just related to the last question, I'm wondering, whether the long-term potentiation that is in dendritic spine, like when there is a burst of this, more proteins are getting synthesized due to plasticity. And could it be in the dendritic shaft region, the long-term replacing proteins are getting down-regulated?
I don't know the answer to that. But I think that you're touching on something which is very interesting, which has to do with the RNA translation being local in the dendrites. And that raises the possibility, you still need tags because the translation is likely to be in the dendrites rather than the individual synapses. So you've still got to capture those plasticity proteins. But it raises the possibility, which is being investigated a lot by electrophysiologists, of quite complex dendritic computation. The computation engine isn't just the cell body, but different dendritic branches, and that can be then controlled by different sorts of inhibitory neurons, somatostatin, cholecystokinin, positive interneurons that are regulating the way in which different dendrites. Now, whether that would extend to long-term depression or not, I don't know.
Thank you.
Thank you so much, sir, for an amazing talk. So I wanted to ask you whether, so we know that there's a very wide spectrum of memory capacities in the population. Also, there are people who we always sometimes call as fast learners or they're slow learners. So there's inter-individual differences or genetic, I'm not sure if they're genetic. Do you think they're at the anatomical level of having more connections for more, more potentiation, or is it the level of, more translation of the plasticity related proteins? So what do you think?
Very interesting question. So at the extremes, for people who suffer from various kinds of genetic abnormalities, this happens in things like autism spectrum disorders, for example, then we think there may well be a genetic basis for abnormalities in the way in which synaptic plasticity is operating. Those could be contributing to the cognitive deficits that are seen. For people who are not suffering from that, what we do know is that there are structural changes happening in the brain that correspond in some way to the particular skills that they learned, whether they're motor skills or cognitive skills. So to give you two examples of this is that people have looked at, on a structural basis, at the right hemisphere in violin players, and they find an expansion of the region corresponding to the digits, which reflects, how they play the violin. So that's one example of the way in which the brain is changing in response to a particular experience something's happening. Another famous example is the work of Eleanor Maguire in London, Irish neuropsychologist, who studied London taxi drivers. And she found that in London taxi drivers who had acquired what's called the knowledge, where they're not allowed to use GPS, they have to remember every street, that they have an enhanced, a larger posterior hippocampus. it's apparently quite strikingly larger. And also, if you look at whether they've been a taxi driver for 10 years or 20 years or 30 years or 40 years, then you find a natural correlation between this expansion and length of time they've been driving. Now, you know what science is like. We scientists were very hard on each other, right? And so everybody, when she published, said, look, it's nothing to do with navigation. It's all those fumes in London that they've been inhaling for 40 years. That's what's shaping their brain. So good scientists, she did the same experiment on bus drivers. You don't see it in bus drivers, even if they've been driving buses. That's nice little example.
Interaction test. So there are individual differences in social interaction.
Which social interaction test do you mean?
The one where you couple study for the memory that they remember to go, the LTP increased after the social interaction and the rats remembered where to go.
Right.
But then there are individual differences in social interactions. Like some rats or mice, they for 10 seconds, something move on and move on. But does it matter that how much they interact? Because there are many diseases where social interaction is included.
Yeah, I don't know the answer to that, to be honest. Part of the trouble is it's very difficult in these animal experiments to control precisely the degree of attention that the animal might be giving at any one moment of time. If you can't control that, you don't know whether 5 seconds or 10 seconds or 15 seconds is all being consumed with common amounts of attention. So it's quite difficult to actually, you know, even though with animal sense you've got much better possibility of having control, it's actually quite difficult to do.
Question that I guess you could have covered in your talk, but that comes up in regular lectures, because HM could remember things like not afterwards.
Yeah.
And how does that stand out?
Yeah. So there are really two schools of thought. One school of thought is that the hippocampus keeps information for a while and then it's all consolidated in cortex. So if you have damage to the hippocampus, as happened with HM because of his surgical ablation of the hippocampus 1954, then information acquired earlier will be stored in the cortex and could be used as access. But a different school of thought reflects a sharp division between episodic and semantic information. And there's a feeling that many of the tests of what's called remote memory that have been done in the past, including with HM, were actually tests of semantic memory and factual knowledge, but not tests of episodic memory. And so it's entirely possible that the storehouse of knowledge that we acquire through our life is indeed cortical, completely cortical, transferred from the hippocampus. But the ability to travel mentally in time, to bring back to mind an experience from the past, requires always the hippocampus. And when people have done tests of autobiographical memory, there's a lot of work suggesting once you have damage to the hippocampus, that's gone.
I don't agree with Marcel. I just want to know, what you mentioned there was a taxation of memory that we need to forget about things. So there are some that people have photographic memories, or there are some rare people that they do not forget anything. So how is that relating to
I
like?
didn't quite follow the question. The people with particularly good memories.
They remember things their entire life. There are some rare diseases. And the people with photographic memories.
Yeah.
The saturation.
Yeah, such patients have been studied carefully, and quite often their life is in some difficulty because of their inability to get things. So how it happens, I don't know. But there may be some fine-tuning of the consolidation mechanism such that far more things can be kept. But they have intrusions all the time which get in the way of ordinary daily life. And so I don't think it's right to say that it's an entirely beneficial state of affairs. I think that's the general view. And the Russian neuropsychologist Alexander Luria wrote a book about a particular patient, and how difficult his life was because of his inability to do that.
Perhaps you are politicians who don't. Get
Yes, we know politicians.
I have an entirely different kind of question.
Right.
You showed Cartier-Bresson photographs.
Yes.
The question is, do you know the mechanism by which he captured these moments? Did he take plenty of shots and he selected the one that's then? Because that must not be having technological possible. How could he capture it? Was it a special thing? I mean, did anybody examine his brain?
Oh, him?
Or is it?
I think it was just his habit. I mean, he was a photographer, and these pictures of Parisian life in the 50s and 60s that he's got. I mean, there's a little boy carrying his van ordinaire back to his grand-mère. But there's a very famous one of a picnic beside the Seine, and lots of other pictures in his books. And I think one of the things that's sort of interesting about, perhaps he had an eye, and what he sort of meant by the decisive moment was not just this business of it happening once, but of it capturing some aspect of French culture. That there was a particular moment that you do that, like the little boy, as it stood at the time. I think there was these two aspects to the sort of decisive moment in his thinking. He was an amazing man. He lived to like 103 or something like that. He's only recently died, I think it's a few years ago.
You know, he had a very interesting India connection.
Oh no.
He was here during independence.
Right.
And also during Gandhi's assassination. Some of his photographs from that transition period are extremely important Indian memories of independence, as I mentioned. So
Yes.
I thought
That's nice to know. Yes.
great job. Thank you.
So thank you very much for being here, and again, thank you for a fantastic.